Okay, thank you for the introduction. Today we are talking about PFAS chemicals and the application of those chemicals or the presence of those chemicals in our industry. I have a presentation for you, which we've titled Beyond PFAS, the Final Filtration Evolution with a New Low-Binding Filter Technology. So please relax and listen to what we have to say. I'm sure you'll find it quite informational. So Sartorius has been around for a long time now, over 100 years. The company was really started in around 1925 with some patents and technology from Richmond Zsigmondy, who won a Nobel Prize for chemistry. In 1927, a company was founded, which is essentially Sartorius Work AG. And this company was around the design and manufacture of filter membranes using the technology from Richard Zsigmondy. In 1979, we developed a filter called Sartopore P using cellulose acetate membranes. In 1995, we developed a new filter called Sartopore using PES membrane. In 2012, we redeveloped another PES membrane, and this was released to the market in a naming of Sartopore Platinum. And this was a modified version of the PES membrane. And then 2017, we introduced a new product of a single-use variety for very large volumes of filtration. So along here, we can show that we've pioneered filter solutions from a very early industry age until the current time. Filtration is really part of our DNA. This is something we're proud of and something that we've worked a long time toward. We're constantly changing to adapt the industry needs. So at the moment, one of the hot topics is really Annex 1 and the spelling out of PUPSIT, so pre-use, post-sterilization integrity testing. So in 2021, we released a specific filtration skid to answer this need for the industry, which is essentially being discussed as we sit at the moment. It is present in Europe. It is also present in the United States and the Asian regions. And here we have a filtration skid that answers this problem. So if you are a manufacturer and you need to do these pre-use post-sterilization integrity testing, it is a reasonably complex task, and Sartorius has come to the industry with these solutions, both in a manual form and also in a automated form. The next big launch for us was really around the next generation of our single-use large capacity filtration options that are designed to be discarded after use, obviously being single use, and moving into larger pharmaceutical plants where stainless steel operations are probably a thing of the past, really, and the requirements are for systems that can be disposed of after use, therefore not requiring cleaning and the associated cleaning validations. So we're constantly looking for the next challenge. One of the challenges we see at the moment is PFAS. So these chemicals that are present in many different industries, but also present in our industry and present in the general environment that consumers exist in. So the PFAS challenge. These are synthetic chemical compounds with multiple fluorine atoms, also known as forever chemicals. They're reaching mainstream attention, I would say, outside of our industry or other industries. For instance, my wife has had discussions on this. She's not a scientist. She's not involved in science in any way, but is acutely aware of forever chemicals and the impact they may have on our health and our children's health. So in our industry specifically, we see PVDF and PTFE filter membranes that are present and are basically these chemicals, and we need to address this at some point. These chemicals are persistent and bioaccumulative and can be found everywhere in nature now, including in animals and humans. So they're starting to penetrate into our food streams, if you like. They're suspected of causing health or adverse health effects, including cancer in humans. So in 2023, the European Chemical Agency proposed a restriction for PFAS as part of a REACH legislation. This is now in the process of being finalized, and we're waiting to see really how this will be administered to the biopharmaceutical and pharmaceutical industries. But you can imagine if there are restrictions put in place, even the suppliers of these chemicals to our industry could become restricted. So 3M, for example, have already announced that they will discontinue manufacturing some of these substances in the near future. And therefore, it could become a supply chain issue if you're manufacturing filters or other products for use in biopharmaceutical with these chemicals present. Similar activities to the European Union have been seen in other regions around the world. So there's a couple of examples here. In Minnesota, the USA, discontinuation of PFAS from 3M has been recognized and well discussed within, I guess, internet forums and the industry in general. So restriction is a limited use or even ban of manufacturing, selling, and or usage of prohibited substances. So, as I said before, this could become a real problem in terms of supply if these chemicals become harder and harder to get, or fewer and fewer are actually manufactured. And therefore, alternatives need to be sought out in terms of how we manufacture our goods for biopharmaceutical or pharmaceutical use. The critical point here that we'd like to draw is PVDF membrane filters. These are often used in final filtration solutions and have particularly good properties, and hence why they're used in those solutions. So we'll talk a bit more about that in a few slides' time. So a couple points down the bottom for future reading. So for more information, do seek out the ECHA and the new regulations that are coming through on these chemicals to verify what I'm saying is actually correct, and to be able to assess your own uses, your own presence in this discussion space. We see here on the right some examples of where PFAS chemicals are used: cosmetics, personal care, fast food packaging, water-resistant fabrics, cleaning products, non-stick cookware. You know, they're quite present in our current consumer goods. You know I have a number of different things that would contain these. My frying pan, you know, some of my waterproof weatherwear. These things all contain these chemicals. So we need to start to find solutions to steer our industries away from that. So what has Sartorius done? Where are we thinking in this space? How have we focused our R&D? So we've accepted the challenge of finding something to remove at least one of those critical PFAS chemicals from the product ranges that you see in the industry for filtration, namely in sterile filtration or liquid sterile filtration. So we've developed a new filter that we've labeled Sartopore EVO. And this is a PES development of sorts, where we have developed a membrane that has a reduced absorption of excipients that we know particularly low absorptive with PVDF membranes. We've named an example here of polysorbate 20 and 80, but this can extend to others as well. It had to be safe and validate bacterial retention, of course, for a sterilizing-grade filter. It had to have a higher filter capacity and flow rate performance compared to the currently used PVDF membranes from some of our competitive suppliers. And critically, it had to have no intentional use of PFAS materials in construction. The PFAS-free statement here could be challenged a little bit because some of our manufacturing processes may have PFAS materials present, but the critical point here is that there is no intentional use of PFAS material or chemicals within the manufacture of Sartopore EVO filters. I've said already what the target application of this new filter is. It's really at the final filtration of a drug product before filling. And specifically, we're looking at MABs, viral-based therapies, and mRNA therapies because of the associated excipients with those and how readily they bind to, say, normal PES membranes that don't readily bind to PVDF in the same way. And therefore, it made sense to develop a PES filter with higher flow rates, higher dirt holding capacity, with lower binding. And that's exactly what the Sartopore EVO has delivered. So it had to be a sterilizing-grade filter, obviously. And then the focus is here really on those protein-based therapies. So I've mentioned already that we developed this PES membrane called Sartopore 2 in 1995. Thing to know about PES membrane or PES material is it is hydrophobic by nature. So it has to have a hydrophilic additive in the case of Sartopore 2 added during the casting process so that it's easily wettable and drug product can go through the filter without any sort of issue. There's no special functionality to reduce absorption, though, critically on Sartopore 2, and it has good wettability due to this hydrophilic additive that's added in the casting process. We then developed Sartopore Platinum, and this is a surface-modified PES. So again, it's a PES and therefore hydrophobic by nature, but we make some hydrophilizing compound, which is cross-linked to the membrane. So a surface modification to make it more wettable. It's even more wettable than Sartopore 2. So the improvement of the PES hydrophilicity is minimized protein absorption, excellent wettability, and moderately reduced polysorbate absorption. However, this could be improved upon. In some slides' time, we show what that absorption looks like in comparison to Sartopore EVO and in comparison to PVDF. So with Sartopore EVO, we now use a post-casting modification and a new hydrogel surface modification of the membrane. And this has the ability to elevate the PES hydrophilicity. So it basically takes what we did with Sartopore Platinum and turns up the dial. There's much less absorption and it's much more wettable as well. And critically for MAB products, it minimizes polysorbate absorption to levels that are similar to PVDF filters. So the available filter configurations are designed to really meet the different market needs. The first filter we've launched, which has been launched this year in Q2 and is available, is the double layer version of this filter. So it has a pre-filter membrane of 0.8 micron with an asymmetric ratio. So you can see here in the electron microscope picture that the top of the membrane, as you flow through from the top of the screen to the bottom, has a larger pore size, and the bottom of that membrane then has a smaller pore size. This gives a nice funneling effect and increases the flow rate for that specific membrane, as well as its dirt holding capacity. And then you notice the final filter membrane has a symmetric portfolio of pores through the membrane, which increases its ability to function as a 0.2 micron membrane and pass those critical bacterial challenge tests. So critically, it's the same membrane structure as Sartopore 2 XLG, and it has this new surface modification to ensure high throughput, but also lower absorption. Next year, we plan to launch in Q2 a single layer version of this same filter, which will have an asymmetric portfolio to the pores to increase that flow rate. And these will really be for applications that are essentially clean already, but still need a 0.2 micron filter to assure sterility of that product. So where are we targeting this membrane, or where are we seeing it as being quite useful for the products in the industry? The first is protein-based therapies. We have really good evidence-based studies on this now that we can share in application notes around protein concentrations, as you can see on the screen, and higher protein concentrations as well. So the critical quality attribute, or the CQA here, is absorption of proteins and excipients. So critically, if the excipients are there to prevent absorption of the protein to the membrane, and they actually get absorbed to the membrane, their job is lost. So if you've got a low absorbing membrane for excipients, this can assist across the whole filtration, particularly at the start. Also for virus-based therapies, so AAVs, LVs, and vaccines. We are currently developing some data for AAVs specific to the use of Sartopore EVO filters and this low absorption profile. But we do have LVs and vaccines that are here as well. And you can see the CQAs are yield and absorption of excipients again for these products at this critical stage of filtration. And finally, nucleic acids, so pDNA, RNA, and LNPs. We have data for pDNA and LNPs, and we're developing data for RNA at the moment. And the CQA here, of course, is yield and throughput. Notice on the right-hand side here, we have a complete manufacturing suite with Sartorius equipment. So going from, say, a freeze-thaw bag through to a mixing system, through to the final filtration on the pupset skid, and then finally across to the right through to a filling line, all in single use. So how does this filter actually perform? We have a number of data sets that we've generated with customers to show the performance of our filter against what we see as the key competitor in this target space using PVDF membranes. So namely, this is the Merck Millipore Durapore filter. That is noted to be in a number of these processes where protein absorption or excipient absorption is a problem, and therefore PVDF has found its best use case scenario. So how does it look compared against our filters? So if we compare the flux rate at half a bar per 10-inch element with Sartopore EVO, Sartopore 2 XLG, which is again a PES membrane, and Merck Millipore Durapore 0.2 micron, you can see here that the flux for the PVDF filter is approximately half that for the PES membranes, either the new Sartopore EVO or the existing Sartopore XLG. If we consider the filter capacity per 10-inch element, and the capacity is measured through blockage rates, if you like, and the model we used was Caro-Ovo, as it's a high blocking fluid and therefore quite representative of a quite difficult process, if you like, in terms of blockage. Here, you notice the throughput is much, much higher for Sartopore EVO and Sartopore 2 XLG compared to the PVDF membrane. Critically, the EVO is almost on par with Sartopore 2 XLG, which is already viewed as an excellent filter for these sorts of applications where you have a high amount of particles that would block a normal filter. So this is really about the membrane chemistry and the known properties of PES that have excellent throughput and excellent flow rates compared to other membrane types. So you can imagine now that when we've developed Sartopore EVO, we wanted these characteristics, but we also wanted the characteristics of low absorption. So we have some more data here with regards to filter capacity and 5% IVIG. So this is an internal evaluation again. And you can see 3 filters plotted, and you might notice that there's a 4th filter, which is another prototype that Sartorius has had, and it's not named here. But critically, you can see that the PVDF filter from Millipore has blocked at a much higher rate than the Sartopore EVO or the Cytiva Prime filter, which is another PES filter, demonstrating that the EVO is on par with that competitor PES filter as well. The initial flow rates here with the same solution are shown to be higher with the PES filters than the PVDF filter, as you would expect, because of this asymmetric pore ratio with the regards to the PES filters. And this is a known property for PES filters, right? So then we must consider, well, okay, there's PES filters from all different types of competitors. So now with Sartopore EVO, we must also acknowledge that it has a low absorption profile. So the next set of data I will share shows what this low absorption is actually like. So looking at the table here, the scale up of filter for milligrams per meter squared, you can see Sartopore 2 from Sartorius. We have a figure of 291 milligrams per meter squared. When we look at the EVO filter, it's 63, which is actually on par with the PVDF membrane from Millipore. We then also tested a PVDF from Cytiva Paul, which gave it 383 milligrams per meter squared, and a PES filter from Cytiva, which was 147. The conclusion here is, firstly, that the Sartopore EVO is performing similar in terms of absorption to the Millipore PVDF filter, but also it's not just about the material itself. If you compare the Sartopore 2 PES membrane to Sartopore EVO, it's got a much higher milligram per meter squared absorption. And if you also compare it against the Cytiva filter, it's also much lower to this filter. But critically, the PVDF filter from Cytiva is much higher than all. So there is some amount of physical nature to how these membranes are cast as well, leading to the overall conclusion that we've shown the new development from Sartorius of the Sartopore EVO PES filter is performing as good as the best PVDF filter. And therefore, you must then consider if PFAS chemicals are a concern for the market in the very near future, that we now have a PES membrane from Sartorius that can perform as good as the PVDF filter. So, so the applications could be changed over to these filters in the case where your regulations come through and PFAS chemicals become harder and harder to obtain. Another data set considering a drug formulation with 30 milligram per mil and 150 milligram per mil of MAB and PS20, polysorbate 20. Here we've compared a PVDF filter with our Sartopore EVO. And you can see that the polysorbate 20 concentration is basically the same for both filters as we take those fraction across the batch. Critically, though, at the first fraction, you'll see the PVDF filter is actually bound more than the PES filter, i.e. the performance is really well noted now through the studies we've done. Some of these studies are available to filter consumers through application notes, and we're publishing those as we speak. Some more data. I'm sure everyone here loves data, right? So data tells a story. Here we've compared a number of different filters to Sartopore EVO in relation to polysorbate 20 in a protein solution and the concentration of polysorbate 20 over time. We've taken some pauses during these tests, represented by the blue, orange, and green squares on the graph, where at a 30-minute pause, after this, you can see some dips from the PVDF filter that are not present for the Sartopore EVO. The Sartopore Platinum obviously is performing lesser. It hasn't been designed for this specific absorption resistance. However, the EVO is performing at the same level as PVDF membrane in this study, and critically, doesn't show the PS20 concentration drop after these critical pauses. So in the end, the PES filter has an overall lower PS20 absorption tendency than the PVDF filter we've tested against. And you can see, obviously, the control that we've used of our own Sartopore Platinum filter, it's performing much better than this. More data. Again, some data generated with our customers. So this is Polaxima F188 in placebo solution and the concentration of PX188 over time. And you can see here we've plotted one of our prototypes that hasn't been pushed through to development yet, Sartopore EVO and the PVDF filter from Millipore. What is noteworthy here is that the concentration fluctuates over time and was shown to do that with each of these membrane types. The performance of each membrane is really on par. So dynamic adsorptions at the start with 30-minute pauses are not noticeable for all filters. So again, we've shown that with this specific combination, our new Sartopore EVO filter is performing similar or the same as the market leader for PVDF. More data, and I promise this is some of the last data I'll share today. However, what we're showing here is that polysorbate 80 in protein and placebo solution are binding much lesser to Sartopore EVO than the equivalent PVDF filter from Millipore that we've designed EVO to work in the applications of. If we compare that then to Sartopore Platinum, our own PES filter that has a relatively high absorption in these sorts of scenarios, you can see this is dramatically different. But importantly, we've got about a 5-fold lesser absorption for Sartopore EVO with this specific polysorbate 80 in protein solution. So again, we're showing a very low absorption of PS80 with the EVO compared to PVDF. This was a 1-hour static slope as shown on the screen. So in a nutshell, the features of Sartopore EVO are the lowest absorption for protein and excipients and high hydrophilicity for easy wetting and integrity testing, which is a critical point to understand. And I'm sure those of you watching this who have done filter integrity tests in the past really appreciate what that means in terms of wettability for a filter. And the technology used here is the new membrane surface modification, the hydrogel, post-casting of the membrane, which allows for these features to be prominent on the Sartopore EVO. The third feature, high flow rates and total throughput and reliable bacterial retention, and also no use of PFAS material in construction, is around this PES, polyether sulfide chemistry, that we've used in these filters. Noteworthy here is also customization options. So Sartopore EVO filters can be ordered as single items or as configured products in customized solutions, as you might see for transfer sets, as we term them, a fairly simple filter, tubing line, and sterile connector, all the way through to more complex systems like pupset systems, pre-use post-sterilization integrity testing systems, where Sartopore EVO can be placed in here with all the requisite tubing and other plumbing associated with those tests. So the product features, flow rate. We have a similar level to our market-leading Sartopore 2 XLG, a high flow rate, in other words. We're at about 95% of that filter. Filter capacity, again, similar to XLG in terms of its ability to hold dirt or to not block, around 95% of that filter. The design is the same portfolio range as Sartopore 2. Critically, though, we have adsorption of proteins and excipients, which are basically at the same level as the market-leading PVDF filter, and 70 to 80% less polysorbate 80 absorption than our own Sartopore 2. And finally, the excellent wetting and flushing, which is similar to Sartopore Platinum. And let's not forget that there is no PFAS material intentionally used in these filters, compared to PVDF filters, where the membrane itself is a PFAS chemical, and therefore potentially will find some issues in the near future due to regulations and therefore supply issues. So the portfolio, how it looks at the moment, and the launch dates. The first set of filters were launched in May this year, and you can see this is really our test filters up to the small scale capsule filters for 210 centimeters squared. In July this year, we launched the rest of the range, really. So from mini cartridges all the way through to our larger filter transfer sets have now been made available with Sartopore EVO for ordering. And finally, in September this year, we expect to have the MaxiCap multi-round or MaxiCap MR available with the Sartopore EVO filters, as well as transfer sets with MaxiCaps, as you can imagine, once that's available. For the MaxiCap MR, it is also made available for the transfer sets. And you can see pictures of this product range on the right if you're not completely familiar with them. At the top are the various filter elements in capsule form, and then you have a pupset skid on the bottom left and a MaxiCap MR on the right. The delivery conditions, this is obviously critical for any end user working in the pharma or biopharmaceutical space. The two broad ranges of filters that you see in use are cartridges and capsules. So with cartridges, they're delivered non-sterile. They're in double pouched PA or PE bags. And the sterilization methods available to these filters are inline steam sterilization, dry or wet steaming, or autoclaving. So you can prepare these filters on site into your stainless steel systems. And the specifications on number of cycles that you can do and the conditions of those cycles are given here. The capsules, midi caps, inline maxi caps, and T-style maxi caps are all delivered sterile. So these will again be double pouched in Tyvek bags. They can be either autoclaved or gamma irradiated. So critically here, though, know that if you autoclave a filter, it is then possible to gamma irradiate. But if you gamma irradiate first, it's not possible to autoclave that filter after that gamma irradiation cycle. So the Sartopore EVO filters essentially have been designed in a hybrid way using PET fleeces for drainage purposes, which allows this duality of sterilization methods. But importantly, we will not offer filters that can only be autoclaved or irradiated. So they'll both of these sterilization methods will be available for any specific filter. So it's more than a filter. Some points on our design focus on this filter. It's designed to increase filter performance, as well as having the lowest absorption of proteins and excipients. It has this high flow capacity due to the PES membrane, and also the best dirt holding capacity due to the PES membrane. And this potentially and likely allows the downsizing of fill and finish filters, which again has a secondary function of reducing waste and disposables within a process. So you can imagine a PVDF filter as a 10-inch. It may be possible to change that 10-inch out for a 5-inch Sartopore EVO filter. Because we're not using PFAS materials here, we can guarantee long-term security of supply. We're not going to be challenged by a world where PFAS chemicals are very likely to become harder and harder to obtain, which would obviously have an impact on manufacturers of filters that are using PFAS chemicals in their filter designs. And finally, we're reducing environmental impact. So by avoiding the use of these forever or PFAS forever chemicals, we're reducing an environmental impact here, and also in relation to how those chemicals, the use of Sartopore EVO to reduce the size of filters can also reduce the amount of waste we're creating. So also the focus here should be that forever chemicals are suspected of causing health problems and must be burned at very high temperatures to completely destroy them. Some of this work was done in my home country of Australia by our CSIRO to show what temperatures exactly are required to just destroy PFAS chemicals. Just a brief slide on where our manufacturing sites are for these filters. You can see 2 locations with pin drops here. This is Göttingen and Yorko. So in Göttingen, we will have all types of filters manufactured as 5-inch cartridges, and in Yorko, only the 5-inch cartridges would be manufactured. The assembly of filter transfer sets could happen in Göttingen, Germany, Yorko, Puerto Rico, Albania in France, and Mohammedia in Tunisia. And you can see now also on this map the large number of sites we have worldwide with sales organization, distribution centers, and application support centers as well. So we really do have great footprint of all potential markets within the planet. Remember, though, that the Sartopore toolbox exists across a broad range of applications. We're not discontinuing Sartopore 2 or Sartopore XLG or Sartopore XLM. We're merely complementing this filter. So the target for Sartopore EVO in terms of where it would be applied to a process is in form and fill filtration, where it's critical that you have lower absorption before your drug product is filled. As opposed to Sartopore 2, where these would be used in applications with medias and things like this, where high flow rates and total throughputs are important and efficient bioburden reduction. Yes, you could use Sartopore EVO there, but the Sartopore 2 already has a number of applications that are validated in this place. As does Sartopore 2 XLG in downstream intermediates where you do require high flow rates and total throughput as well. So the Sartopore toolbox, we offer Sartopore filters for any biopharm manufacturing step that requires a sterilizing-grade filter to ensure high efficiency with excellent throughput and flow rate using a versatile membrane material without the intentional use of PFAS. In fact, any amount of PFAS in there would be a challenge to find. It would be residual from our manufacturing processes. Our manufacturing processes are under review at the moment in terms of where we may have some of these low slip or anti-slip sort of chemicals for manufacturing processes that would be changed in the future when we find alternatives. So complementing the Sartopore EVO and Sartopore 2 filtration portfolio are our filter transfer sets. There's a picture here of a PUPSIT manual system, which is used to basically answer the concerns of PUPSIT and really the spelling out of PUPSIT in Annex 1 and now the pressure from many regulators around the world on implementing PUPSIT in processes that haven't previously had them. In the middle, you can see our Sartocheck integrity tester. So this has the latest quality risk management and data integrity standards, which are industry leading effectively. So these are also critical elements of Annex 1, and this is something that is really influencing the regulatory space and the regulators around the globe, including in Asia and North America and obviously Europe. The next point here is an automated version of PUPSIT. So having in the past myself been involved in the rollout of a PUPSIT system prior to working with Sartorius at a manufacturing site, I can say that an automated version would have made my life much easier in terms of the number of errors that were found during integrity testing and the delays that were seen, therefore, in batching because filter integrity tests had failed. Not the filter had failed, but the integrity test had failed due to leaks and other handling issues. So you can appreciate looking at these systems on the screen. There's a number of different valves that need to be operated in the correct sequence to be able to do the integrity test. So appreciate here that a automated system would allow for those integrity tests to happen with the functionality of having much, much, much lower rates of false fail tests. And finally, our validation service that we name Confidence. So this lab that we have around the world, and this is replicated in many different countries, which allows the critical bacterial challenges, chemical compatibility, extractables, leachables studies, and integrity test value generations for customers with their product against our filter base. So critically here, we're offering a special offer at the moment in regards to any validation service for Sartopore EVO. If that work needs to happen at the quickest possible time, we are removing the expedition fee for those works. So this reduces the normal lead time of about 10 to 12 weeks for studies to be performed down to 6 weeks. So quite a decent offer there. So that's it for the slides today. I thank you for your attention and appreciate your consideration of Sartopore EVO and how this looks in a world with changing regulation around PFAS forever chemicals and what that might mean for our industry in the very near future. Thank you for your attention. We'll now open the floor for any questions that might have come up during the presentation.